Hi students, welcome to Year 11 Biology and Module 2, Organisation of Living Things. In this video 14, we're going to overview mammalian digestion. Now there's a lot of aspects to this that we can cover in a little bit more detail in class, but what I want to try and do is just touch on some of these key aspects of the digestion of foods in a mammalian digestive system, and that is physical and chemical digestion, the absorption of nutrients, minerals and water, and the elimination of solid waste. So the first thing we should do is have a look at the alimentary canal. The alimentary canal is basically just a one long tube that starts at your mouth and ends at your anus. And it's a complete tube, and into this tube are attached a number of different types of glands and organs that secrete different materials into that collection of food that you swallow, and all sorts of different processes happen, both physical and chemical processes, to break the food down, to basically select which things are going to be absorbed, and then to release what's left behind. Obviously everything's going to start from the mouth, and that's where the food enters the alimentary canal. We have teeth, we have a tongue, and we break down mechanically a lot of the food that we consume before we swallow it. That goes into the esophagus. From the esophagus, food travels to the stomach, and often it'll sit in the stomach for some time. And there are a number of different mammals that have certainly more stomachs than we do, and who allow that process of digestion to happen for much longer than we do in terms of how long food spends in our stomach. From the stomach, the food will pass to the upper part of the small intestine. There's three parts of the small intestine, including the duodenum, jejunum, and ileum. And this is really where the fats are first started to be broken down, but also where we have a lot of absorption. Some very important nutrients and organic compounds are absorbed through the villi, through microvilli, little tiny finger-like extensions on the epithelial cells that line the digestive system or the alimentary canal, there to facilitate absorption of nutrients via their very large surface area to volume. From the small intestine, the food will then pass, or whatever's left now of the food will pass into the large intestine, which is the colon, and we have an ascending, a transverse and a descending colon. From the colon, and the colon is the site primarily of water reabsorption, then into the rectum where food is stored and then expelled through the anus. So let's have a look at each of these kind of key components of mammalian digestion one by one. And the first thing we need to look at is physical digestion. So basically physical digestion is breaking down large pieces of food into smaller pieces. It's a physical change, not a chemical change. So it doesn't involve any chemical reactions. And often what it's trying to do is to do this very important biological concept, and that is increase surface area relative to volume, which means that we get our enzymes, which are actually involved in chemical digestion, to be more efficient. So before we get the enzymes to work, we want to try and see if we can reduce that amount of food that we have from large pieces into smaller pieces, and in so doing increase the surface area for the enzymes to work on. For examples where physical digestion might occur is most obviously in the teeth, and we have a number of different specialized teeth. We have incisors at the front, a kind of nippers. We have canines, which are good for and common teeth for meat eaters, and we have premolars and molars, and these are our good grinding teeth, and they are primarily for this process of mastication, which is breaking food down into smaller pieces. So that process of chewing, which is very important. It's important that we don't try and swallow our food as whole as possible, that we actually try and leave it in the mouth long enough for it to break down into smaller pieces. Now once we swallow, we've got something which is called a bolus, and this bolus then moves from the mouth to the stomach through the esophagus via a process known as peristalsis. And this is a muscular squeezing, basically, which which is such an incredibly powerful squeeze of the bolus from the mouth to the stomach that this would still happen even if you were standing on your head. I don't know why you would want to eat standing on your head, but if you did, your food would still reach your stomach against gravity because of this strong peristaltic movement. Once food reaches the stomach, there are stomach muscles around the lining of the stomach that can contract and relax and will mix the stomach contents together. Now, there are also enzymes and hydrochloric acid that's actually introduced into the stomach, and that's part of the chemical digestion process. But these contractions and relaxations are designed to help squeeze the food around, mix it up, continue that process of physical breakdown into smaller and smaller pieces, increasing the surface area each time. And another area of physical digestion is the action of bile. So bile, which is produced in the liver and stored in the gallbladder, can also then be secreted into the upper part of the small intestine, where it'll act on large lipid or fat globules and emulsify them. That means it just turns them into smaller lipid globules. The second of these processes is chemical digestion, and chemical digestion basically involves a chemical reaction. So when we have an actual physical change in the chemistry of what we've been eating, then we've moved from a physical digestion process, sometimes called a mechanical process, into a chemical process. And most of these processes are a form of decomposition. So they're going from large molecules to smaller molecules. And the simplest ones to think about are things like carbohydrates, carbohydrate digestion of something like starch, which is a complex carbohydrate, a large polymer molecule. It starts digestion in the mouth. So an enzyme called salivary amylase, and we know that this ASE ending is very common to enzymes. And so a salivary amylase produced in the saliva, it's something that is secreted into the mouth when you eat, and it acts on starches and turns them into simpler sugars, so the glucose, the maltose. So if you want to have this little mental picture, say think about eating a piece of bread which is high in starch, hold it in your mouth long enough for the salivary amylase to start breaking that starch down into maltose, and you'll actually get that slightly sweeter taste as a result of that. Of course, most of the time we don't notice that because we eat our food so quickly. In the stomach, we have proteases, and as I said, there's not just enzymes that are secreted in the stomach. There are also hydrochloric acid, because these proteases, like pepsin, for example, require a very low pH for them to work optimally. And remember, when we looked at enzymes, we talked about optimal function, and that is usually a temperature and a pH, and potentially substrate concentration and cofactors, or the absence of inhibitors that are going to ensure that the enzymes are working at their optimum. Now, proteins are made of amino acids, so once again, this is kind of a decomposition process as we break proteins down into the smaller amino acids, which can then be absorbed. As well as the bile that helps to emulsify fats, there are lipases that are produced by the pancreas that are also secreted into the upper part of the small intestine, and they're going to be acting on lipids to reduce those lipids to monoglycerides, to fatty acids, in order to, again, make them easier for the body to access them. And occasionally, and certainly for humans, but particularly for a lot of other animals like ruminants, think cows, for example, the microflora is critical. It's critical for us because bacteria in our gut or other microorganisms in our gut help to break down some of the material that we eat that we can't break down with our own body chemistry. And often the taking of things like antibiotics can wipe out some of these very good bacteria, and hence you see these ads sometimes for replacing the good bacteria when you've been on a course of antibiotics. This is a form of fermentation, and it's specifically acting on cellulose, which is another complex carbohydrate. But unlike starch, we don't have the enzymes to break down cellulose. So we have to have a bit of assistance. We have to have this microflora, these bacterial fermenters, and some organisms are very, very big, and they have either ferment in their foregut or in their hindgut and keep food in there for very long periods of time to extract all of the nutrients they possibly can. The third thing we need to talk about is absorption. And absorption happens through microvilli. And villi are these tiny little finger-like extensions that we see lining the small intestine. And they are designed to increase surface area. So we're talking here about an increase in surface area to volume, which we know means more efficient exchange. And so this is where the smaller molecules, some of the water and the mineral salts are obviously small enough. If you drink water, you don't need to digest that in a chemical sense. That water will automatically, through osmotic pressure, move from your digestive system across into the epithelial cells and then into the bloodstream. Salts can do that as well. So some of the ions that are present in mineral salts, think calciums or sodiums or chlorides, they can automatically migrate straight across as well because they're so small. But organic nutrients like complex carbohydrates or proteins are too large, and so therefore they have to be acted upon in order to reduce them in size, chemically split these long molecules into shorter pieces, and those smaller pieces can then be absorbed through the microvilli and go into the blood system. And again, this happens through the process of diffusion. So we create a concentration gradient, an area where there is high concentration of something, and that's going to move to the low concentration that we have in the cells and in the blood. Absorption happens in both the small and the large intestine, though in the small intestine it tends to be more of the nutrients, and in the large intestine it tends to be more water absorption. So keeping everything reasonably moist helps to keep it moving. That process that we talked about, peristalsis, continues to help to squeeze the food through the digestive system, and so that we can try and maximize the amount of extraction of nutrients that we possibly can. So the alimentary canal is basically this tube that is where we put all of our food. In order for us to get any benefit out of that, we have to absorb those important nutrients from the alimentary canal through these microvilli or through the lining of the colon, the large intestine, into epithelial cells, into the little cells that line the digestive system, and then from there into the blood system. And the blood system obviously is our transport system, so it's going to move everything around the rest of the body. Finally, something we don't want to talk about for too long, but it is part of this process, and that is that not everything we eat we can use. So therefore, after we've carried out physical and chemical digestion, after we've carried out nutrient absorption via diffusion, and we've removed water from through the colon from what's left, then we now have something that's relatively solid. And this solid waste that we can't use basically moves into the rectum. And once it's there, nerve signals are sent to the brain that begin that process of eliminating the solid waste. So it doesn't happen straight away, but there is a little warning that it's kind of time to clear this load. And again, it's peristaltic movement that brings the waste through the rectum and exits it out through the anus. Now, this is kind of a very quick overview of the key components of mammalian digestion. Obviously, depending on the diet of different mammals, they are going to have more or less complexities, or at least we see differences in some of these important structures that are part of the digestive system that are based on whether it's, say, a nectar feeding habit, an animal that feeds on, say, insects, termites, or ants, whether it's an animal that eats meat predominantly, whether it's an animal that is eating very low nutrient grasses and so therefore needs to extract everything possible from them, or an omnivore like ourselves that can eat a range of different types of foods. So there are different types of mammalian digestive systems that go with each of these different types of diets, but we might have a look at some of those in just a little bit of detail in class. Thanks for watching.